Thank you for the invitation to be here. My daughter goes to Havergal College, and when she heard I was speaking at TED Med again, she said, "Oh, that's really cool." And I am honored to be here. It is very cool. But then she said, "What are you going to talk about?" And I said, "Well, you know what I do, ex vivo lung and lung transplant." She said, "That's boring. They've heard that already." So I said, there might be some people who think that it's cool still and haven't heard it, but I promise I'll change it and show you some new stuff. So when you talk about people dying of lung failure, we talk about lung repair, replacement, and regeneration, the 3 R's. So replacement is what we do today, transplantation and artificial lungs. Your lungs are broken, we give you new ones. Artificial lungs are an attempt to mimic what Mother Nature does. Can you make a lung that'll exchange gas and work as a lung for you? But can we repair your own lungs? That's the promise of regenerative medicine, and then we won't need any of this replacement. But for now, what I'm going to show you is repair of slightly used lungs to make them better so that you can use them for transplantation, and also how we can take damaged lungs and regenerate them and repair them to build new lungs. So lung transplantation is really a very exciting Toronto story. In fact, November 7th, 1983, was the time that they did the first successful lung transplant in a patient in the world right here at the University of Toronto at Toronto General Hospital. Hercules there was a monkey who got the first double lung transplant successfully in the research lab by Joel Cooper and his team, and they brought this clinically to make lung transplant a reality. Part of the problem was figuring out how do you actually take lungs out and put new ones in. And the other part of the problem was how can you preserve lungs that are so fragile and easily damaged, take them out of the body, carry them across the country and put them in someone else. And part of my master's thesis project was to develop this lung preservation solution called Perfidex, which allowed us to preserve lungs like that. They're filled with oxygen while they're being transported, and they're kept cold. And the idea of cold is that you shut down the organ. So you slow down the dying process, you slow down the need for metabolic substrate, and you allow the lung to just basically wait it out and you rush and do your transplant. And I'll get back to that concept. So in 1983, the first single lung transplant was done here in Toronto. In 1986, the first double lung transplant, replacing both lungs, also done at Toronto General Hospital in 1986. And another world first, 1988, the first lung transplant for cystic fibrosis in the world done here. And since those early days, we've continued to increase the number of lung transplants we perform and decrease the mortality or risk of the operation so that it's a pretty routine operation now at Toronto General Hospital, doing over 100 transplants a year. In fact, we just finished doing a baby lung transplant this morning at Sick Kids. And lung transplantation really has allowed many people to get back to normal and active lives at work and play. All of these people would have died without a lung transplant, and you can see the kinds of things that we've got them back to after a lung transplant. Our problem is there aren't enough donor organs, and you can see that this is the total number of organs in the red line that are available in the Ontario region, and these are the number of people waiting for organ transplants. These are the people that actually get the transplant. So what can you do to help these people for the here and now? Artificial lung technology is one of those things. So this is a 1971 picture of the first patient on an artificial lung in California. The artificial lung is this whole room of equipment. And this thing, the size of 2 garbage cans, is actually the gas exchange membrane that was used at the time to keep that young trauma victim alive. Go to 2000, Toronto General Hospital. Here's the artificial lung, patient flat on their back, basically anesthetized, but much improved technology. Now you get technology like this. This is the Nova lung, fits in your hand, and that's your artificial lung. In fact, it can even be run so that you use your own heart to pump across the membrane, and you don't even need a pump. And basically, it does the gas exchange technology. And now we've got systems like this. This is the Nova lung with a pump in it if you need a pump. And the patient can actually walk and wheel their lung along with them and be ambulating and not flat on their back on a ventilator. And technology continues to evolve where we will soon get portable, fully implantable lungs that will be a replacement. We can keep people now for 6 to 8 months, almost a year, on an artificial lung. So it's not perfect. There are a lot of complications to it, and we usually use it as a bridge to transplant, but it's getting there. This is an example. This girl, we were doing CPR on her when she had primary pulmonary hypertension, and we put the Nova lung in. This was 2 days later with her dad in the intensive care unit at Sick Kids. She stayed on that Nova lung for a month, and that was 6 months after her transplant, and this is several years later after she graduated from college. So taking the next step is, how else can we help these people? Can we repair lungs? And I showed you that the way we currently preserve lungs is to keep them cold so that we actually slow down the dying process. And the problem with slowing down the dying process is you slow everything down. So the lung shuts down, and its ability to regenerate, its ability to heal, your ability to facilitate its healing is also slowed down and impaired. And most importantly for lung transplantation is you only find out how it works once you plug it in and turn it on. And if it doesn't work, you're in big trouble, your patient's in big trouble. So that really hinders our ability to use lungs that might not be okay, and so we don't. So we developed a system here in Toronto where we could put a lung in a chamber, ventilate it with a breathing machine like a patient in the intensive care unit, use an artificial heart pump like the ones we use on cardiopulmonary bypass, except we use it backwards. The pump goes across a membrane which removes oxygen, adds carbon dioxide to the circuit, and then the lung can do its function of removing carbon dioxide and adding oxygen, and therefore we can assess lung function over time. And we've made it so that we do everything that we can to protect the lungs so that the system doesn't add damage. And so what we've done now is taken this cold static situation to a warm, alive, 37 degrees normal body situation where we can assess the lung, diagnose it, treat it, confirm the effect of our treatment, and then know that we're transplanting a known product with a predictable outcome. And this is what the system looks like. These are human lungs that were not usable for transplant, and at the time of this video they'd been on this circuit for about 8 hours, and they're breathing. We can measure their function, we can measure the vascular resistance and the airflow and so on. Here we're looking down with a bronchoscope, and we're using it to deliver gene therapy into the lung to genetically modify the lung to make it work better after transplant, to make it more like self so that your body doesn't reject that organ. So we can manipulate the organ before we even put it in and confirm that it works before we put it in. And we have the luxury of time to treat the lung as a patient, personalized medicine for the patient. What's wrong with that lung? What treatment does it need? Has it responded to the treatment? Are we okay? And we brought this to the operating room at Toronto General in 2008 and performed a clinical trial that was successful and published in the New England Journal that this is really a new era in transplantation, personalized medicine for the organ. So you can bronchoscope the lungs, and just like we bronchoscope patients, looking in there for diagnosis, for treatment, as I showed you. You can take x-rays of the lungs in the operating room, and you can see how they improve over time. And you can do tests on them. And we've done arrays of genes, and have found biomarkers that predict whether that lung's going to be good or is going to have a problem. And if it is going to have a problem, can we make it better after transplantation? We're also developing gene chips to look at what they do over time on the ex vivo perfusion so that we can monitor in real time the response to the therapy. We're working with people from the University of Waterloo to look at metabolomics. This is a little catheter that you can slide into a vein, but we also put it into our circuit, and you can look at metabolic products in real time as we're treating and perfusing the lung. A very important thing is that most of what you've heard about personalized medicine and molecular diagnostics is being developed primarily in the field of cancer. And if you go to your oncologist, you get your tumor biopsied, you come back in a day or two and they've got your tumor profile and they decide on the personalized medicine approach to treat your specific tumor. That's 2 days or 1 day. And you know, for cancer treatment, that's probably okay. For transplant, it's not. So where we're focusing is we need the gene profile in 10 minutes, not in 1 day. So that is our challenge. We're working with Shana Kelly here at the University of Toronto. She's developed a nano chip with these little nano hairs that have the gene sequence on it, and your gene anneals with the hairs on the chip, and you get an electrochemical readout in a few minutes rather than 5 or 6 hours. And so that will help us in terms of really directing our treatment. We're looking, as I mentioned, gene therapy using adenoviruses, the common cold virus. Take all of the genes out of the cold virus, put in genes that we're interested in to repair the lung, to modify the lung, use the virus, which is very smart at getting into your lungs, as you know, to carry the gene into the lungs and express genes that will repair the lungs. We've done this in human lungs and showed that we can improve lungs that didn't perform well and make them perform well so that they can be used. We're also looking at, can we make genes expression last longer in the transplanted lung? These rats had a lung transplant where the gene was upregulated by treatment at the time of transplant, and you can measure the gene expression by a bioluminescence camera, and we've shown that you can have this gene expression from the one course of treatment at transplant for up to a year after transplantation. And you can turn these genes up and down as needed to control rejection or not. So have the lung actually make its own immunosuppression locally as we continue to build lungs that will be able to handle the challenge of transplantation. Taking it one step further, if the lung is so broken, so damaged that you can't repair it or augment it, can we actually build new lungs? And one of the ways that have been looked at is take the actual lung itself, remove all of the cells so that you have the scaffold, and then add back the cells that are you. Use your stem cells to build a lung for yourself. And Harold Ott and his colleagues had done this with lungs, and you can see here, these are lungs, rat lungs that are being decellularized. So when you remove all of the cells, you see this pale white structure that is the scaffold of the lung. He added back the endothelial cells and epithelial cells on the right side of the membrane to make a lung, and then they went ahead and transplanted the lung. Now that lung only worked for about 20 minutes. That's not a home run yet, but you can see the concept. We'll get there. And in fact, Harold and his colleagues just recently, you may have heard, did the same thing with a kidney, made a kidney that worked for a little while. So the whole field is moving in that direction. So how does our Toronto ex vivo system fit into this concept? This is the ideal bioreactor, or the beginning of bioreactors, that if you're going to grow a lung, where are you going to keep it until you know it's a lung, until you know it's working as a lung, or as you're repairing it? So Tom Waddell in our group, who's working on things like this, is looking at, okay, it's fine to decellularize a rat lung that's the size of your thumbnail. Well, what about the lung the size of a human lung? And this is a pig lung. The decellularizing fluid has some black dye in it so that we can look at the distribution as you're perfusing the lung to make sure that you do decellularize the whole organ effectively without damaging some parts more than you need to. And part of it is we may have to develop a rotating bioreactor for distribution of the fluid, because the lung is very delicate and more pressure isn't necessarily better. What about other parts of the respiratory system? This is a patient of mine who needs a new trachea from her larynx down to the carina right in the lung, and there is no prosthesis for this. So can we grow a new trachea for a patient like that? So we're working on developing bioreactors to take decellularized windpipes or tracheas. There's a human trachea that's had all of the cells removed out of it, so you can see it's pale white. And then Tom and his colleagues have developed a system to have a media on either side of the airway that are ideal for the airway cells on the inside and the mesenchymal stromal cells on the outside, and have this bioreactor grow a new trachea. But the important thing is the airway has to be stiff. When you take a breath in, you make a negative pressure, it would collapse. You have cartilage in your airway that prevents the collapse. So they put it on CT scanners and apply negative pressures, and you can see this airway totally collapses with very little airway pressure. So how can you make an airway that preserves the cartilage, or can you grow new cartilage to mimic the structure that Mother Nature made? The airway is lined with epithelial cells. epithelial cells in our airway are lined up in a way, and they have little cilia that beat, that carry the mucus up and carry any foreign bacteria and pathogens up so that you can clear your lungs. When you grow cells in a petri dish, they just grow all higgledy-spiggledy like this, and they're not going to be a functional laminar flow lining for the airway. So what we've done is taken plates with little nano grooves on them. And when you grow those cells on the nano grooves, you can see that there's the higgledy-spiggledy ones. On the grooves, you can start to line up cells and slowly build an organ that has its functions. The organ isn't just a pile of cells in a lump. Each of these areas of the organ have specific high-end functions that we have to figure out which are the ones we need to mimic. And when you think about this, this is not the future, it's now. Many people, there's the lung system. We're working on a liver system at Toronto General Hospital. Others are working on kidney and heart. And in fact, we have the first organ regeneration laboratory currently working at Toronto General Hospital. OR 18 is our organ regeneration laboratory. And the other day, in 2011, a colleague of mine from Chicago had a patient who was dying, and he had lungs that weren't any good. He said, "Can you repair them in Toronto, because the FDA hasn't approved this yet?" So we flew the lungs to Toronto General Hospital, repaired the lungs, and transported them back to Chicago, where he transplanted them successfully. So I'd like to close by saying that there's a large number of people in the Latin or thoracic labs at Toronto General Hospital Research Institute that contribute to this fantastic work. It's a great time working. We're doing very exciting things, as you can see. And the most exciting thing about University Health Network, where we work, is that we have a clinical team in lung transplantation that really have the sense of adventure, passion, and commitment to save the lives of patients with very challenging problems and allow us to bring what we discover in the research laboratory to the bedside. Thank you very much for your attention.